Hendra virus infection dynamics in Australian fruit bats. The well-known Hendra virus emerged in 1994 in Australia. At the time of the article being published, there has been 7 noted human cases and 45 equine cases of the disease. The virus infects horses and humans via a wildlife reservoir. In the case of Hendra, this is through fruit bats, also commonly known as flying foxes, like the ones pictured. These fruit bats are a natural host of the virus and are asymptomatically affected by the disease. To provide further information on the infection dynamics of the Hendra disease, a 3-year longitudinal study was undertaken using reverse transcription polymerase chain reaction, RT-PCR. This technique is commonly used to detect gene expression levels, in this case the Hendra virus, genome in pooled urine samples that were collected from flying fox roosts. The objective of this was to investigate infection occurrence and frequency in flying fox populations and to investigate the genetic diversity of the Hendra virus in flying fox populations. 3 roost sites were identified in 3 geographically differing sampling locations, all greater than 1,500 kilometres apart, in southeast Queensland, far north Queensland and the Northern Territory. These locations were visited every 4 to 8 weeks where possible. In the late afternoon at the selected roost, plastic sheeting was placed under the tree in which flying foxes were roosting. The following morning at sunrise, pooled urine samples were collected from each and held on wet ice. Depending on the roosting density, each sheet could hold urine from an estimated 5 to 20 individuals, giving a sample size of 50 to 200 individuals from 10 sheets. A total of 1,672 pooled urine samples from 67 sampling events were collected and tested between the 1st of July 2008 and the 30th of June 2011. Numbers varied with year and location, with 311 samples in Year 1, 465 samples in Year 2 and 896 samples in Year 3. There were 138 samples from Southeast Queensland, 320 samples from far north Queensland and 214 samples from the Northern Territory. 25% of the sampling events and 2.5% of urine samples yielded detections. The proportion of positive samples was statistically associated with year and location. These findings indicated that Hendra virus excretion occurs periodically rather than continuously and in geographically dispersed flying fox populations in the state of Queensland. The lack of any detection in the Northern Territory suggested prevalence possibly varies across the range of flying foxes in Australia. These findings suggest that flying foxes can excrete virus at any time of the year and does not correlate directly with seasonal clustering of the Hendra virus incidents in horses and associated humans. This is that 70% have occurred in June to October. Therefore, the identification of these factors will greatly strengthen risk minimisation strategies for horses and ultimately humans.